The next speaker is Dr. Cristianne Rijcken, founder and CSO of Cristal Therapeutics in Maastricht, to talk about CriPec nanomedicines, the translational path from preclinical to clinical studies.
Thank you, and first of all, thanks for the organizers for giving us the opportunity to present. And actually today the focus of the talk will be, and I slightly modified the title, is more focused on the tunability and the manufacturability of the CriPec platform. And tomorrow afternoon I'll present an update about the clinical program that we are performing. So for those of you that don't know me, my name is Cristianne Rijcken. I'm the founder of Cristal Therapeutics, and Cristal Therapeutics is a small company based in the Netherlands, where we are developing the CriPec-based platform. We ourselves are focusing predominantly on oncology, but together with our partners we also work on the development of CriPec-based products in other areas than oncology. And the way how we do that, as I said, it's a kind of a hybrid model. The broadly applicable platform, either ourselves product development or in collaboration with partners, and we do that with venture capital money. So CriPec, what is it standing for? It stands for kind of a dual effect, namely that we have nanoparticles that are initially stable in the bloodstream, stable meaning also being inert, so really having a long residence there. And then ultimately accumulating in either the tumor tissue or in other chronic inflammatory diseased area, whereby ultimately the effect is that we aim to have solely the release of the compound at the target site and having less exposure to the healthy tissue, so really shifting the balance from therapeutic efficacy balance to more improved safety profile. The underlying principle is, as I said, the CriPec platform, and this is really the core of the technology, which is that we make nanoparticles in a 2 or 3-step process, where the first step is conjugation of a linker to the API of choice. Then we combine it with a proprietary polymer, and then you get a nanoparticulate structure formed in which the drug as well as the polymer, they're all covalently bound. So in contrast to, for example, liposomes, which are all a physical encapsulation or a physical mixture, this CriPec nanoparticle is one big macromolecule, as everything is covalently linked to each other. And as said, potentially a third step can also be applied, because to the surface we can also conjugate all sort of modalities. So as you can imagine, such a nanoparticle has a different biodistribution over time. What is key is that only the native drug is being released as a result of chemical hydrolysis. So we can use different type of linkers, so thereby really governing the release kinetics of the drug. And in time, the native particle itself is also disintegrating into smaller fragments. So actually, the talk of today will really focus on the tunability, so I won't go over it in detail, but we'll really optimize the applicability of the platform and all its details. And as what you can imagine is this platform can be used for a variety of compounds, either to use existing compounds, and that's what we do ourselves, so we use docetaxel as a compound to improve the efficacy-safety ratio, as well as for some more novel compounds which were formerly not capable of being delivered, which is the oligonucleotide program. And in collaboration with partners, we work in the immuno-oncology arena as well as outside oncology. But maybe first of all, touch upon the applicability for type of APIs. I said broadly applicable, but what does it mean? It means that in recent years, actually in recent decades, a lot of different therapeutic modalities have been developed. So, of course, starting off with the small molecules already way back, they have some challenges, but then in recent years, the proteins and even more recently the oligonucleotides, they also have their challenges for application. And we think we have, with CriPec, a tool to optimize it, maybe not to completely be 100% effective, but at least to, for example, assure more specific tumor targeting, or to increase the stability of oligonucleotides, and even also more specific tumor cell targeting. So to show you the example of docetaxel, where we entrapped the docetaxel compound in small nanoparticles, 65 nanometer sized, and then we compare the tumor uptake as compared to Taxotere, so equimolar levels, then you see a really significant higher uptake of these nanoparticles as compared to the native API. And not only a higher uptake of the nanoparticles, but also a sustained release in the tumor tissue of the released docetaxel, so the active ingredient is really leaking out of the nanoparticle over time. Not only have we applied this to small molecules, but I said also for novel APIs. So this is the same approach that we use for, in this case, siRNA, where we conjugate our linker to an azide-modified 5' antisense strength, And this type of linker is then used, and I just want to elaborate on the modalities that we use. So this is for the cross-linking in the nanoparticle, and this is then the acid-sensitive bond. So ultimately we generate upon acid-sensitive hydrolysis, we create a compound with a small modification, but we have assured that this does not affect the potency of the compound, as ultimately the antisense strands has to be affected in the risk loading. But this approach has been successfully made, meaning the conversion, the conjugation is very efficient, which is important for such a costly material as siRNA. And then we were capable of entrapping this siRNA linker into nanoparticles of around 55 nanometer size. So actually the entrapment of the siRNA does not affect the particle size, and also with a fairly good entrapment efficiency and loading capacity. But even more importantly, the release kinetics are really acid sensitive, so that means in the lower pH we have a much more faster release as compared to physiological conditions. So the concept here is, as you can imagine, fairly good long circulating in the bloodstream, and then only in case of uptake into the tumor cell having selective release over there. And in last month we have also demonstrated that these nanoparticles upon intravenous administration to tumor-bearing mice really result into long circulation in the bloodstream. So as compared to the native siRNA, which is rapidly eliminated, as is known to all of us, we do see a really long circulating of the nanoparticle, which in turn translate into much higher uptake into the tumor tissue, ultimately so really to be used for also therapeutic efficacy. And as I've shown you, a small molecule, but also now oligonucleotide, we dare to say that this CriPec platform is broadly applicable for any type of API. So that's what we're currently exploiting. And maybe then to touch upon a couple of the tunability features of the CriPec platform. So the tunability holds true for the particle size, so fully governed by self-assembly, and actually the polymer length that we use to make these nanoparticles fully governing the ultimate particle size that are generated. So this is for CriPec empty, so without any drug in there. But of course, even more importantly is that if you entrap a drug, that this correlation also still holds true, so full control of the particle size, while at the same time having very nice low polydispersity, so indicating the good uniformity. Then the other parameter is the linker release. I said fully tunable by the type of linker, and that's what is shown in this picture, that by using different type of linkers, and I won't go into the chemistry, I can elaborate with you on it later on, but only changing the type of linker that is used, we have full control of the different release kinetics. So that's regarding the content of the nanoparticles, but then what you can also think of one API in a nanoparticle, actually what we demonstrate that we can also entrap two APIs in one single nanoparticle. And in this case, you see nicely two different release kinetics. And as I've shown you in the former slide, we have different linkers that we can use, so we have full control over release kinetics. And this is where we see the application in diseases where you like to target different pathways, but each with a different release kinetics, ultimately aiming for synergistic efficacy. Then regarding the surface modification of the nanoparticle, as I indicated, so of course this is not to scale, so this is the nanoparticle that is presenting with an A site to the surface. And this example shows where we conjugate an RGD as an active targeting moiety to the surface, while that ultimately results into this, via click chemistry, into this triazole bond being formed. And the reason why I indicated this in blue is that in fact that we make nanoparticles with have a 15N enriched azide moieties onto the surface. And that allows us to follow this type of chemistry by a 15N NMR, meaning that we literally can follow the conversion of the azide present on the surface, and thereby really can quantify the number of ligands that have been conjugated to the nanoparticle. So it's not like a black box, well, we add a certain amount of ligand and let's hope it's being conjugated, but we can literally follow and monitor the quantification of that. So the former slide was about an active targeting moiety, but actually what we would also like to monitor the biodistribution of the nanoparticle in preclinically, but ideally in the patient setting, and ultimately going for patient stratification, so only treat those patients which have an enrichment of the CriPec in the tumor tissue. So this is a European Horizon grant that was awarded to us, where the simple approach is that to conjugate a PET label to the surface, more specifically zirconium, as it's having a long decay time, and ultimately only treat those patients which do have this enrichment of the tumor tissues. So the underlying chemistry is alike in the former slide with the RGD as an active targeting moiety. Here we use actually a so-called BCN desferal. So BCN is the click reagent, and the desferal is the chelator, which ultimately can conjugate the radioactive zirconium. So alike, we conjugated to the nanoparticle triazole bond being formed and monitored. And then just before patient administration, we complex it with the radioactive zirconium and then can give it to the patients. So this is a very elegant approach that we, from a chemistry perspective, developed. It took us quite some hurdles, but we have successfully developed it by now. And even more importantly, what we've shown in a preclinical study in tumor-bearing mice, where we monitored the fate of either the non-labeled, so the plain material, as well as the zirconiumly labeled material, that the PK profile is equal, and even more importantly, that also the tumor accumulation is equal. In other words, this modification does not affect the biological profile. So this was really, for us, a very important route forward, as we are now actually treating patients with this and evaluating the PET scanning and thereby monitoring the biodistribution of the nanoparticles, particularly the tumor uptake. Then very last two slides on the manufacturability, because I've shown you all the different features of the CriPec platform and its tunability, but ultimately it comes down to making material that can be evaluated in a clinical setting. So I thought I'd show you actually the process, how we make ultimately the zirconium CriPec docetaxel. And let me announce that we made this all possible together with a CMC producer, the GMP producer, ChemConnection, which is a company based also in the Netherlands. And actually, the zirconium CriPec docetaxel is only a 3-step process. More specifically, it comes down to these 3 steps. So first of all, we have to make the nanoparticles, so we have the polymers on the shelf, then we make the nanoparticles micelle formation followed by immediately cross-link, so this is one pot synthesis followed by purification. The second step is then making the conjugation of this desferal to the nanoparticles that are available as bulk drug products, and then ultimately it's of course need to be filled and finished into vials. And as said, just before patient administration, we don't do this ourselves, of course, because we do not have radiochemical capacity, but this is done at the VMC, so the party that actually performs the clinical study. So we're there in their radio facility, perform both the radio labeling, the purification, and then ultimately the patient administration. What we've demonstrated in recent years is that particularly the tangential flow filtration is very effective for the purification and concentration of these type of nanoparticles. And even more importantly is that the upscaling is very straightforward. So we can make material on very small scale, and that's what we do ourselves for the early stage studies. But we have also successfully upscaled towards big scale production up to 40 liters at ChemConnection site. And then the very last slide, which is about the QA/QC, as also by the former speaker. Without going into really a lot of detail, we have clearly had to monitor all kind of parameters to ultimately release the clinical batch. And actually what we've done is develop initially the methods ourselves in-house, because the commercial CROs, they're not so well experienced with nanoparticles. So we initially developed the methods in-house, then transferred them for validation at the GLP facilities. We've demonstrated so far that all the clinical batches produced are compliant to the set specs, so meaning the production procedure is robust, and even more importantly, the IMPDs have been approved, so meaning that the regulatory authorities are also in line with our reasoning and have accepted the material produced so far. So with that, I'd like to thank the public for listening, and I'm open for any questions.
Thank you very much. Questions, please?
No?
Yeah.
Yes, a quick question is, you have really nice control over the size. Did you see differences in tumor accumulation based on that size, because the surface properties seem to be the same?
Yeah, of course, that's a relevant question. We have not done the studies in depth, but clearly we base there on literature that the smaller the better. So that's why we're really focusing toward 30 to 50 nanometer size particles. But we have not done a very exploratory study ourselves where we really evaluate the effect on tumor uptake.
One more question.
How many years did you use from R&D until you had the product in the market?
Well, in market, that's a good question, because we're not there yet, but we are about to start phase 2 just later this month. From all the way, I'd say that it was formerly my PhD work, so that was back in 2007, and 2011 we established the company. Then we came up with a decision for CriPec dose to excel, so thinking out loud, it's 7 years from concept to phase 2 start. So I'd say that's fairly reasonable.
I would also like to ask a question. So what kind of deliverable dose would you see to the brain feasible? Is it like single milligrams, micrograms, or?
To the brain, you mean?
Yeah, in the best case mode.
Right. I would say that for the brain, you really need to have active targeting. So there, I think it would be feasible, but to what extent, that's a clear, good question. I'd say in terms of tumor uptake, we're percentages per injected dose per gram tissue. If we could come up to 1% injected dose in gram brain tissue, that would be a major achievement.
Mm-hmm. Thank you. So thank you very much, Cristianne. And now the next